Today, yeah. Okay. Okay, so the main topic for today, or what Klaus usually teaches today, is statistical mechanics of proteins. So I'm going to just teach or show you guys what he usually teaches. We're going to have the group picture today at noon. Also, we're going to have a coffee break again when we're done with the first part around 10:30. And this afternoon, again, from 2 to 6, you guys work on the tutorials. Similar to yesterday, you can keep working on the tutorial you were working on yesterday, or you can pick a new tutorial. So let's get to business then. So I know some people are familiar with the molecular dynamics. How the molecular dynamics simulation process. So I mean, I know people here who is familiar with molecular dynamics. Who has the, okay? So maybe half the people here. So well, for those that are not that familiar, what you usually need is you need a starting structure, right? And how do you pick a structure? How do you decide which structure you're going to use? Like, you just go to the PDV, you go to a friend and ask them to build the model. How do you usually start with a structure? That people have ideas?
Some kind of experimental data.
So like experimental data, like you have X-ray, right? You have other sources of data. How about you ask your friend and you get a model? Would you trust that model? Not necessarily, right? So the first thing is starting with a good starting structure, and it's very important that is a good model, because your results are going to be highly dependent on the quality of the starting structure. That doesn't mean that you have to disregard anything that is in silico, right? The structural models are as valid, computationally derived models are as valid as experimental models, as some experimental models nowadays, so long as they are carefully built. So what are the typical or the most used source of structures has been solution NMR and X-ray crystallography, right? And depending on the resolution of that structure, you can use some other software to do refinement, right? There is a variety of software you can use to refine those experimentally derived structures. So now, in the more recent years, there has been the development of the cryo-EM technology. So does anyone here work on cryo-EM or is familiar with how cryo-EM has been shifting the field? Like high-resolution cryo-EM and just like regular electron microscopy? Okay, so cryo-EM has been shifting the way that structures are being derived, because if you notice, for instance, on the top of the graphic, we have the size of the typical size of the complexes or the proteins that can be resolved by the particular method. So if you look at NMR, it's usually limited to very small proteins, right? And this is widely known in the field. If you look at X-ray, they can actually access larger and larger complexes. But cryo-EM actually can look at subcellular components. They can look at like an entire protein assemblies. So what has been changing over the years is the cryo-EM used to be this, they used to call it blobology, because you can see only these big blobs. You couldn't see really atomic resolution. And this has been shifting over the years. There has been both in hardware and software, there has been a lot of development in how the data is processed. And they can actually reach like up to like, nowadays they can actually compete with X-ray in terms of resolution. They can go below 3 Armstrong, which is not really typical at the moment, but you can, if you go do a survey of every Nature science paper that has cryo-EM on it, it's about 4 Armstrongs right now. So it's competing with X-ray. What's typically done in terms of computation is to combine, use the computer to combine NMR, X-ray, and cryo-EM, and use different packages like the molecular dynamics flexible fitting, which is the one we develop in our lab, Direx or Sidus, and combine these multi-resolution structures into a single molecule, into a single structure. So there are many sources that you can use or you can look for a good starting structure. So
can you also combine with NMR data?
it depends what kind of NMR data. So like for instance, if it is like NOEs, those are relatively trivial to combine. If you have, I mean, we're working on combining chemical shifts, which are a little more involved to incorporate in the calculations. But in principle, yes, you can also combine solid state NMR. So you can, I mean, along what you're asking is you can incorporate, for instance, if you're doing an MD protocol, you can combine them, you can incorporate them as restraints. So for instance, if you have NMR, you can incorporate those restraints as distances restraints. So like you don't allow certain group of atoms to deviate more from a particular predefined distances. And you can also use even FRET, like a fluorescence probes. So you can also use FRET distances as restraints in the simulations. So this is commonly done nowadays, is to incorporate, like, actually, even if you have an NMR or an X-ray structure, it cannot be in a biologically relevant state. So you can actually optimize that starting structure using MD and informing the MD simulation with other experimental restraints that can guide your molecule to a more biologically relevant state. So there is also a subset of techniques that are what are called enhanced sampling, which allow you to overcome barriers. Like normally you are kind of stuck into a single basin. We'll touch base more into that later. But there is also the in silico structure prediction that can either be using ab initio structural prediction, kind of what Rosetta does. You just give it a sequence and then you get a predicted structure. Or you can also do homology modeling. So which are people familiar with homology modeling? Okay, so for those who never done homology modeling, is you have your sequence of interest, and then you survey the PDV, the protein data bank with your sequence, and then you look for candidate structures that have some sequence similarity to your sequence of interest. You can actually quantify the sequence similarity. You can have either identity, which tells you like identical base pairs that are conserved in the sequence, or you can have sequence homology, which is a different score of how the two sequences align. So then using that structure in the PDV, you use that as a template. You essentially copy coordinates, and then you apply some scoring functions and some minimizing of the coordinates, and you generate a new model for a particular sequence. And in general, homology model is accepted to generate reasonably good models that you can later refine using, I mean, you can, if you don't know anything about your model, maybe you're working on a human protein, but there is only a bacterial model available, so you can create a human homologue using the known bacterial structure. And then you can use some other refinement techniques, like perhaps you have more data from NMR or FRET that permits you to either evaluate the model, validate the model, or you can also look at how like do something like cross-validation, where you predict a model, you don't use any of the experimental restraints, and then you see how much of the predicted model actually overlaps with the experimental restraints. Something that you should be very careful whenever you get a model, even if you just download it from the PDV, is to do a structure evaluation, right? You should check secondary structure is in place. So this is even before minimization. You should make sure that the secondary structure doesn't have any problems, because that later on when you're running the molecular dynamics simulation, any secondary problems, any secondary structure problems that you might have had is going to result in serious problems during the simulation.
Did you attribute that mostly to this error, or?
Sorry?
Would you attribute that mostly to error, or why? I mean, why would so many PDB structures necessarily have the incorrect secondary structure? I mean, what might some of the
So for instance, if it is a X-ray, right? If it is a low resolution, you know, like how X-ray is pretty much a person looking at a density and assigning atoms manually, right? So there is a chance for error there. Yes.
right? They're just reporting the data.
Yeah. So they're actually so the PDB is actually an interpretation of the X-ray data. Right? So there's still room for error, and although, I mean, if unless it is like 1.1 Armstrong, which is really high resolution, if you're in the 3 Armstrong regime, there's still a lot of room for a lot of wiggle. In
How do you check for the secondary structure? Do you look at the structure and say, oh, this part looks off?
so, for instance, if the typical example is a kink that is missing, right? So like if a helix should have kinked and it's going straight, things like that. Because like kinking of a helix is something that is going to take a while in a simulation, and if you don't have the kink in place at the beginning, it may be a problem. You can also have like beta strands. They're also very hard to fault or like actually converge in a simulation. So if it's a little bit off, you can actually it can break apart and things like this. In any case, it's very important to keep track of secondary structure. Something that we do is also perform a cluster analysis. And this goes along the lines of stability assessment and we'll touch base on this later on again, is cluster analysis is during the simulation, you're actually sampling multiple different conformations of the protein, even with slow variations. So really to assess which of those structure is, you actually have to look at all of them and see what the differences between like how they cluster in time and see which one kind of built a probability for each of those substates. In any case, for MD, you have to be careful with what structure you start with because your results are going to depend a lot on your initial structure. Yes?
So you just do a visual check for secondary structure?
So there are programs that allow you to do a secondary structure check, but also, okay, so here's the other important thing is you just don't, people have this idea that you just throw anything to MD and you don't really have a scientific question, right? So like, I guess at the beginning, you need to have a scientific question to actually start to do the MD. So that means you have a pre-knowledge of your system. So that means that you probably know when things can be, if like something structurally with your protein could be something that is associated with the intrinsic properties of that protein or is just a fluke. So the secondary structure, I mean, there are programs that can tell you if how much it deviates from a perfect secondary structure, but also you should be, I mean, you have to wonder if that might be something real related to the function of the protein.
I noticed in the DMD tutorial they talked about some software package called, I think, Stride.
Stride, yeah.
To evaluate the secondary structure. I didn't look at the article too much to see how they actually did it.
Mm-hmm. So there are different kind of secondary structure checks. Oh, Stride is one of them. There is also the SSP. And also, like, if you go to like what crystallographers use, like Molprobity and all these web servers, they also do a secondary structure check. So, for instance, if you're gonna like helices, they typically have certain angles, and they don't deviate too much, like the sci-fi angles. So you can actually use something like Molprobity and see how the helices are, like how imperfect the helices are. Also, if you look at the header of the PDV, like most newer crystals, they tell you what the features of the crystal are. So what might have been a guess, what the deviations from standard measurements are. But I think in general, the idea is that you should have a good structure to begin with. And that's very important. So going back to the regular, yes?
Sorry, can I ask one question? So since the MD can calculate the interaction between those atoms or molecules within structure, if I start with a bad homology structure, can I expect the good structure after the MD simulation?
So yeah, that's kind of, many people do that. You start with a predicted model, but then you have to use enhanced sampling for that. You have to do a lot of sampling, and then you have to also do a lot of analysis to validate that the model that you found is the right one. So it's possible, but you can derive a good quality model, but it will require a good amount of effort. It will require you to do use an enhanced sampling technique, and so you're at least you can confidently convince your colleagues that you're converged, and you also can convince your colleagues that the structure that because you have an ensemble of structures now, so you also have to convince your colleagues that the structure that you picked is the relevant structure. So it's possible, but it requires, it's not just like you build a homology model, then you press a button, run MD, and then you get a better model. It requires a couple more steps, but it's possible. And I mean, this is, we published this paper this year in Annual Review of Biophysics, and we have a very lengthy discussion in how to use MD to generate better models.
Does that go over a little bit more about what cluster analysis tools you actually used?
Well,
I noticed in the AMD package they recommended something in Vega ZZ, and
so actually I'm writing right now a new clustering for VMD. It should be available in the next release. So, but I'm not gonna, I mean, if we can talk more about clustering, because it's a whole it's a whole topic. But I think that what's important is that you realize there are multiple sources for structures. You cannot disregard in silico prediction of your structures, but you have to be careful how you validate and how you evaluate the quality of your model. And if you're really serious about the simulation you're running, you should probably do this for every system you're interested in. Any other questions up to here?
Are you going to talk a bit more about the enhanced sampling?
Very briefly.
Yeah.
Yeah.
No, because you didn't mention anything about how the coarse grain MD comes in.
So the coarse grain MD is, I'm not going to talk at all about the coarse grain MD in here, but it's probably more related to, actually no, I do talk about it in a little bit. So we'll just wait and I'll talk about that. Let me think. Yeah, we're actually using mostly of the, at this point in time, the coarse grain MD for anything that has lipids, not so much for the protein. So like, right now we're building a whole HIV virion. So we're building the entire, the entire vesicle. So that the lipid composition and like the distribution of the lipids on the surface of the vesicle is very important. But lipid diffusion is very slow. So what we do is we equilibrate the vesicle with a coarse grain model, and I'm going to show something that is similar, but not exactly the same. And then you do reverse coarse grain. So you achieve a close to a real distribution, and then you do reverse coarse grain, and you go back to all atom, so you can start asking more fine questions. But I mean, again, if you're interested in looking at how MD helps to or assist in solving structures that you can later on use for more scientific endeavors, you can check this with this paper we just published. So I think at this point, I think it's very important. We're right here. It's really important that you pay a lot of attention to the structure you're starting with. So the next step is to build the environment of the protein, right? It can be water, it can be a lipid membrane, it can be, I don't know if you have some weird solvent, but it's important that you also perform this step carefully. In any case, once you have the simulation box or you have your system of interest. You simulate using your local cluster or a supercomputer. In general, better off using a cluster than your local, than your desktop, just because of performance. And then you analyze. You use VMD for analyze. You do some pretty pictures, and then you just publish, right? So this is the pipeline to go from a structure to a publication. Yes.
So I was in the lab earlier yesterday, and the building stage, we had a water sphere and a water cube. So I want to know, like, the environment is the same for both. So how does it make a difference in, like, the timing of building when it's equilibrating, whether it's a box or whether it's a sphere? How is that?
How that makes a difference?
Yeah.
So when you have a box, you have periodic boundary conditions. So you have the cube, right?
Yeah.
So whatever comes out of one end of the cube comes in the other end of the cube. In the sphere, you don't have that property, right? You just like have a droplet, right? So I'm gonna talk more about that, but it's related to the electrostatics and how you actually calculate the electrostatics. But
Surface effects.
But,
yeah, but I mean, if you have a sphere that is big enough, you don't have.
The reason I was finding it really counterintuitive, it's not like it's an isolated box, right? When the protein is there, it's in that medium. So if the medium is water, I was wondering if that's the only thing that should matter, the box or the sphere that you're simulating.
Oh, the ions too, right? So you're going to have the water, the ions.
Yeah, but like the geometry, how is that making a difference?
The geometry is not going to make a difference in terms of the dynamics of the protein. It's going to make a difference in the performance of the simulation. So, for instance, well, I mean, right now everyone is working with cubes, but you can also have hexagonal simulation boxes that are like when you cut the edges, you reduce the size of the simulation box, and you can get better performance. But in principle, the properties of the dynamics of the protein should be the same. Yeah. Mm-hmm.
One question.
Uh-huh.
So when we're building this determination by the user of the box size, right? Now from a little experience I had, run some simulations and the box size was small, so I had my membranes actually my cells interacting with others in another box. Is there a way to determine the box size?
Okay, so you have, can you repeat it again?
Yeah, I have membranes or my cells in one box interacting with the next periodic boundary. So is there a way we can determine the box size for perfect simulations to avoid such interactions with?
So your solute is a micelle,
Yeah.
and then you don't want it to interact with the next one, right? Common practice is to have like 20 Armstrong padding between the solute. Well, some people say it's 5, but I mean, it's better to have more padding the longer the simulations become. So I don't know how much padding do you use? Do you just put it?
I just determine the box sizes.
Yeah, so you should probably have, if you have your solute, then you should probably add a little padding of water between the solute and the edge of the simulation box, yeah.
Aren't there also some cutoffs that you can set for the electrostatic interaction?
So we'll get to that later, yeah. So, okay, so in any case, two things that I like when Klaus gives this lecture is, first you start with a scientific question, and then the last thing you need a story to tell, right? Otherwise, you're not gonna get, I mean, maybe you're gonna write a student report and not really a scientific paper, right? So it's very important that when you plan, and it's also very important that you realize that this is not like pressing a button on a computer. It's actually an involved process. So when you start your simulation, when you're at this stage, when you're picking which structure to use, it's important that you have a scientific question, because you're actually looking to make a scientific contribution that is going to have hopefully some impact in some field, or well, your subfield of interest, or maybe everyone is going to be crazy about your contribution. But in any case, it's very important that you just don't think that you can, I mean, MD can be think of as a computational tool that you can just, is a computer doing the work, but it's also important that you realize that the story that you tell at the end has to be of value and not so you just don't run any simulation for the sake of running simulations. So I think the story here is very clear. You make sure your structure is good, you have a scientific problem, and then at the end you have a scientific story to sell, or like a sales pitch. Make it interesting. So I'm going to show a few equations now. We're going to go into the mechanics of how MD works. So I mean, you don't have to understand everything, but it's good if you get a good understanding of how the inner parts of the code works. So what effectively doing in MD is very simple. It's classical dynamics, but just because it's simple, it doesn't mean it's easy, right? So you can think, the first thing you can see is that you have an energy function that depends on the coordinates of your structure, right? So that's why your structure is so important, is because it's determining this energy function. And then anyone who's taken some physics has seen this equation, right? What is that equation? Newton's equation, right? So very, very simple equation. It's a second order differential equation, and we have the forces, but the forces can be derived as the derivative of the potential function, right? So what you're going to be seeing, what you're actually seeing here is that you have a dependence of the position of the second derivatives of the coordinates is going to be determined by the derivative of the potential, right? So the initial, as I said before, the initial coordinates are obtained, well, typically from crystal structures, but velocities, and I shouldn't have this here yet. Velocities are, I'm going to tell you later what we do with the velocities. Okay. So here's where it becomes really interesting. So I'm pretty sure a lot of people here are familiar with your calculus 1. Like, you know, any student can tell me what's going on in this equation. It's a Taylor expansion, right? So it's a Taylor expansion, and what this you have the first, second, and the first order, second order, and third derivative and second derivative, right? So if you do the Taylor expansion and then you do it for t plus some delta t, so like some increment in time, and you do it for another like the past, like you go back in time, another delta t, and you add these two, right? You obtain what is called the Verlet algorithm, right? And it's actually pretty simple. So, okay, let's do an exercise here. So there is a velocity hidden in this equation. I mean, it's not really hidden; it's in plain sight, but it's not explicitly written as v. Can you spot it in the bottom equation? No. So we can split this in two contributions, right? Like this is RT plus RT. So let's forget the first one. So you have RT minus delta t. What is this thing? That's a velocity, right? So Verlet, all Verlet is telling you is that you can actually, if you know your current position, the velocity at the previous step, and you can derive the accelerations from the force field, you can actually determine where the next step is going to be, right? That's all Verlet does. So is that clear? I mean, I think it's a neat trick. Is that clear? So there is. So then I'm going to ask you guys. So we get the coordinates, or better, we get these coordinates t from the crystal structure or previously determined protein structure, right? So how do we get this RT minus delta t? Any ideas? So we have the coordinates, but also, I mean, if we split these two RT, we also have a velocity, right? Which is so in principle, I mean, so we're going to need some velocities, but the velocities we can actually assign them randomly, right? And that's what that's why you let the system equilibrate. So RT comes from the crystal structure. The velocities you just sample from a Maxwell distribution, and then you randomly assign them, and then you just let the system relax. And then you have this other term here that is the potential, right? So it's a very simple, I mean, you just go from Newton's equation, a simple Taylor expansion, adding the two Taylor expansion, and then you have the velocity Verlet algorithm, which is the one that is mostly used in any MD code out there. Any questions about this part? So what do you think this mass dependence is going to do to the dynamics of the system? So what is going on here? What happens when you have a big mass? How big is this term? It should be small. If you have a small mass, like a hydrogen, what happens? So it contributes more, right? So actually, this mass is the reason why you actually have to use very short time steps, and we're going to go into that in a little bit. So yeah, because effectively, if you notice, like this RT plus delta T is going to have a direct dependence on the mass of the particle of interest. So if you have a hydrogen, so let's check our chemistry. So what is the typical oscillation of a CH bond? What is the period of that oscillation? Anyone knows that from the top of your head? It's 10 femtosecond. Yeah. Yes. So it's 10 femtosecond. So in order to be able to sample that oscillations, you actually have to run a tenth of the time, right? So that's why the time step is 1 femtosecond. Is because you want to be able to sample that CH oscillation in a. I mean, this is all. I mean, let's look at this from another perspective. This is an stroboscopic view of the life of a protein, right? So it's like when, I don't know if these stroboscopic lights are still a thing in the clubs, but you have these like 1 second, right? And then you just see these snapshots, right? So this is the stroboscopic look in the life of an atom. So if you are sampling too infrequently, you're going to be observing non-realistic, or you're going to be observing a different distribution of the motion of these atoms. But if you sample with the right frequency, in this case 1 femtosecond, you're going to be able to reproduce these oscillations of the CH bonds. So that's when you, okay, so now that we talk about these CH bonds, so for the people who have actually run MD simulations, how do you actually get to run 2 femtosecond simulations? What's the trick?
Restrain the vibration of hydrogen.
Yeah, exactly. You remove that degree of freedom, right? So like the CH bond is not allowed to oscillate in the 10 femtosecond frequency because it's rigid. We don't have that oscillation. So therefore, we can actually use a longer time step, like 2 femtoseconds, right? And that's what. So it reduces the system size of algorithms that do this kind of analysis, that do this kind of.
Did you say that if you sample
Effects.
too frequent, that it becomes long?
No, less frequently.
Okay, then can I go like down to femtoseconds or so for sampling, like less than femtoseconds?
Yes, but that's impractical.
Don't they do that in vacuum when you're doing?
No,
Okay.
no, at all.
So what happens if we sample it 2 femtoseconds and if we allow the CH bond to be valid?
What do you think will happen? I'm asking you.
We will not sample the 2 states of the bond.
Yeah, and what would that lead to? It actually becomes unstable, right? So you because you are actually you're sampling least frequently the CH motions. So you're actually, the sum over all those motions is not 0 anymore. It adds to a constant. And then you're doing this iteratively. So you're actually, it feels like you're adding energy to the system, and you're shifting it away from equilibrium. So it actually becomes unstable. It actually explodes.
So that's what the rigid bonds?
Rigid, exactly. That's what the rigid bonds parameter in NAMD does, is it freezes that degree of freedom.
And the hydrogen in the water, are those bonds always rigid, or is that all?
They should also always be rigid. Tip 3 water is parameterized with a rigid bond between the hydrogen and the oxygen. Yeah. So even if you're using 1 femtosecond, you should still make sure you have rigid waters, at least for tip 3P. So for tip 3P, which is the watering charm and many other programs, and unless you know you're using a different water, that's a rigid water. So the oxygen and the hydrogen, you can use, even if you're using one, you still need to use rigid water. So the one is for the protein. So you don't. So okay. So you can constrain because actually you're removing the degrees of freedom. You can constrain the hydrogens for the water or for the hydrogens for water or protein. You have the option to in NAMD, right? Now, even if you constrain only the water and not the protein, you still have to use 1 femtosecond. The only reason when you will actually don't have rigid water is if you are using not a tip 3P water, which is a rigid water, right? And and you will know if you're using a non-rigid water.
So if I'm using tip 3, the hydrogens are always rigid?
Yes. Otherwise, you're going to have some problems there.
Is it those extra columns at the end in the PDB file that tell when you choose the rigid bonds, it chooses the that last column to say, okay, this should be a rigid bond?
That's the occupancy I think you're talking about, right? The beta and occupancy. Yeah. But those are for restraints. So those are not rigid bonds. Those are positional restraints that fix those atoms to particular points in space, which they're different from restraining the.
So how are the bonds made rigid inside of the simulation?
Yeah, that's a good question. So actually, that's what there are a couple of algorithms. There are Shake, there's Links, and what they do is they directly remove the degrees of freedom from they're holonomic restraints in the Hamiltonian. So you actually remove those degrees of freedom from the system. They're always coupled. So it's not a restraint, it's a constraint because you're removing those degrees of freedom from the Hamiltonian. Okay, so we talk about the velocity varlet. So are there any other questions?
Will there be any disadvantage of restraining the CH?
You can use 2 femtoseconds, you get better performance.
Yeah, we'll get speed up, but will that be a disadvantage otherwise?
No.
Like in terms of that actual finding the function or dynamics of the system?
And it should be the same as one. Yeah.
For the bonds, if they're not rigid, if you actually have like a vibrating bond, are you modeling as a classic spring?
Yeah. So we're not modeling them. We're using. Okay. So let's talk more about the functional form of the force field potential, right? So we talk about how you need a potential function that depends on the coordinates of all of the atoms in the system of interest, right? So let's look at the bonds. So each of these balls is an atom, and the line connecting them is a covalent bond. So the way that they're classically modeled is as a harmonic oscillator, right? So the way that the force field works is more of like a database, and then depending on the atom, there are multiple atom types. you have to obtain multiple parameters. So for instance, there are 2 parameters for a bond. You have the K constant and also the R0, the equilibrium. You also have 2 parameters for the angles. So an angle is like, so bonds is between 2 atoms, angles is between 3 atoms, right? And then you also model the angle between these 3 atoms as a harmonic oscillator. And again, you introduce 2 new parameters. And for dihedrals, so you have a set of 4 atoms, right? So you have this solid angle between this plane here and this plane here, right? So for dihedrals, you actually have a different functional form. It's not quadratic, but you also introduce a couple of extra parameters. These terms here, these 2 lines here, are commonly referred as the bonded interactions, and the bottom line is commonly referenced as the non-bonded interactions. So what I'm going to ask you guys, what is this term here? Coulomb term, right? That's the electrostatics. And what is this one here? Dispersion, right? The dispersion term. So these 2 terms are pretty well characterized from physics. You can very much, everyone is familiar with these 2 terms. Everything else in purple is a heuristic, or it's a model, and it comes with 3 parameters. So chemists over like 30 years have been working on developing the different parameters for this potential function. If you look at the charm force field, if you look at the amber force field, all it is a database of like that has been collected over several years that you can actually put those values inside this potential. So are there any questions up to here? Nope. Okay. So I want to, okay, there's a question here. So this is assuming that the particles are point-like particles, and we're talking about an atom. Do you think the electrostatics of an atom is that of a point-like particle?
It has electronic degrees of freedom.
Right, that this is only true in the approximation when you completely disregard the electronic degrees of freedom. So recent development in the lab has been focused on including not explicitly those degrees of freedom, but accounting at least for the polarization of the electron density, right? So if you have 2 charges here, so well, we have our typical potential, but if we have 2 charges, then we are not going to start treating them as point-like particles. We actually associate another massless charged particle, which is called a Drude particle. The Drude particle, what it does is it accounts for any polarizability due to the presence of a nearby charge. And it has, well, the Drude particle, the Drude force field, that's not only account for polarizability, but it also includes explicit lone pairs, which are completely absent in any classical force field. So these are 2 interesting features about the Drude force field. It has been available in NAMD since 2013, but something that we, or I particularly find very interesting, is that you break these isotropic electrostatic interactions, which is what we're seeing in all classical simulations. Is it doesn't really have any sense of direction. Hydrogen bonds doesn't really have any sense of direction. Not with point-like particles. So you actually have an isotropy of the electrostatic interactions when you actually account for polarizability.
And this allows for flexibility. I mean, where you're not saying this is a particular system, but you can use it for a variety of proteins.
Sorry?
I mean, the idea of this force field, I mean, you have a set of parameters that works for essentially all proteins, or all.
Work for most proteins.
Or most, really, in reality, most proteins should work well.
Yeah. So it's a transferable, generalizable force field.
I mean, my understanding is a lot of groups, though, will customize their.
A lot of what?
A lot of groups are developing their own force fields for particular systems, too, as opposed to this very general force field.
Well, that's Yeah, well, I guess some people do that. I guess it's better if it's more general, and you're not just like trying to reproduce some experimental structure or things like that. Because, yeah, here we don't. The assumptions are relatively soft versus if you're actually tailoring a force field to a particular problem. In any case, so what is interesting about the accounting for polarizability is the introduction of explicit lone pairs and also the accounting of any anisotropic effects. Yes.
So how does this polarizable force field affect the performance?
Okay, that's here.
Oh, okay.
Yes. So yeah, that's a very good question because I mean, as you might have noticed, you actually, for every heavy atom, you add a new particle. So that means your system is going to grow in size, right? There is another caveat with this. So we talked before about the CH bond, right? And we talk about how what was in that velocity verlet, the term at the far right, what did it depend on? It was the mass, right? But you must think, oh, these guys are crazy, because these guys are actually adding a massless particle, right? We're adding a massless particle. The Drude particle is a massless particle. So you're adding a massless particle. What do you think? And we talk about how the hydrogen, the mass of the hydrogen is limiting the time step, right? But now you're adding a massless particle. What is the time step now? Is it going to go to 0? So actually, you have to do a lot of tricks. I'm using the word tricks, but you have to do some theoretical developments in such a way that you can still use realistic time steps. So you have a massless particle. So like the typical time steps for a Drude simulation are anything between half a femtosecond to a femtosecond. So in the lucky cases, you can go to a femtosecond. In any case, so you're adding more particles. You're no longer allowed to run 2 femtoseconds. So it really takes a hit on performance. So for instance, this is a simulation of a water box. The red particles are the oxygens. The green particles are the Drude particles. And you can see how the Drude particle comes out of, like perpendicular to the, you can see it there, right? Like it comes like out of the oxygen atom. So that displacement is what is accounting for polarization. But I mean, you shouldn't be scared of using the polarizable force field if you feel like, because the performance is actually reasonably well, is pretty good. And more importantly, if you have access to a supercomputing, the scaling is also very good. So it requires more computational resources, but if the problem is well worth it, you should definitely be able to get the performance you will get otherwise with a classical force field. But there is a hit in performance. Yeah. It doesn't support GPUs yet, so you're limited to the. I'll correct that. There, because of the instability of the Drude particle, you actually use something which is called a hard wall potential. So you essentially are limiting you're not allowing the Drude particle to go too far. So that hard wall is not supported in the GPU version. If you don't use the hard wall, it is supported in the GPU version. But that's more of a technicality. In any case, we recently used this force field in a paper we published this year, and that was to study the interactions between the capsid and cyclophilin. Cyclophilin is a human protein, and we tried very hard to stabilize this complex with the classical force field, and it was impossible because at the center of the interaction there are water-mediated interactions that are, we all know water is highly polar. So when we used the polarizable force field, we were able to not only to stabilize the complex, but we also discovered a novel interface at the tip of the cyclophilin. And in case without going into any details, this against This again support my previous claim that if your problem is worth the time and the supercomputer time, if you have a good scientific question, and it really depends on using a more sophisticated force field like the Drude force field, you should definitely not be afraid to use it and explore it as an option. So,
I didn't understand how the massless Drude particles, how was the time step only doubled? Half.
To 1. Actually, normally you go to 0.5. So these are massless particles, but because they're massless, they move really fast, right? So what we're going to touch base on that later on the Langevin equations. So actually, for practical reasons, you end up having 2 thermostats. You have one thermostat for the real atoms, let's call them that way, and you have another thermostat for the Drude particles. So that's a way you stabilize them, and that thermostat is set to 1 Kelvin. So they tend to be unstable because they have no mass.
But in the equation, how is it solved? Putting the mass of 0 there.
Oh, there? Well, that's the here, right?
Yeah.
Like how do you have a massless force? Yeah, we don't. They don't really behave following this same equation here. They're a little different. We can talk later, and I can explain you more how that works. But yeah, it's a good point that they're not treated as classical particles.
And this, did you say, is this built into the current version of NanoD?
It is, yeah.
In 2.10 or 2.1?
I think it's been there since 2.9.
And how do you turn it on?
So, well, it's not only turning it on, you have to build the whole system. So the best way at this point to build the system is there's something called the Charm GUI. And then you can, if you have your Charm 36 PSF and PDB, you can, they have something which is called the Drude Prepper. And then you just upload your PSF PDB. It has to be very well equilibrated. And then you get the Drude PSF and PDB. They actually, at this point, I think they also give you the NAMD configuration file, so you can just keep running on the simulation. But it only works from Charm 36 to the Drude force field, and your system has to be very well equilibrated. Anything that is a little bit, if it is a little bit off, you're going to end up having problems. It's a very delicate force field. Are there any other questions? Okay. So let's go back to the statistical mechanics component of all the molecular dynamics. So these are your, if anyone, I mean, I have statistical mechanics class, you always come across the ensembles, right? And then you have constant energy, constant number of particles, constant energy, constant volume of NVE, constant temperature, constant volume, constant temperature, constant pressure, right? So which of all these ensembles do you think is the most realistic for a protein or a simulation?
It actually depends whether you're doing, well, commonly NVT, but
MVT?
equilibrating when particle are unlocked, so we do the NPT equilibration to make sure that the density of the particle of water is stabilized before we go up. So when running the production dynamics, NVT, but set of equilibrations, NPT followed by NVT.
So if you have a protein in a beaker, it's an NVT.
Open blast conditions.
Right? So okay, so what you're telling is partially true, in the sense that you want to reach a volume where the pressure is more or less what you will expect in experimental conditions, which is 1 atmosphere, right? And then for performance reasons, you can switch to NVT once you've reached that equilibrated volume. So why do you think people don't run simulations on constant energy, constant number of particles? Right, it's very artificial, but still this constant energy, constant number of particles tells you a lot of information about how the molecular dynamics actually works, right? So let's talk about how you keep how do you do the temperature control. So this is the famous Langevin equation, right? Are people familiar with the Langevin equation? Is for a particle in a fluid, and it effectively is telling you that if you're moving in a media, you're gonna have some viscosity, you're gonna have some resistance to freely diffuse, right? So what this is telling you is that the accelerations have this force term, but also more important is this second term here, right? So what this is telling you is that the drag that you feel is going to depend on the velocity that you're moving, but also you have this other term, which is a random Gaussian process, right? So what you're effectively doing is, if you're moving too fast, because of the sign of this term here, you start extracting energy from it, right? So if the velocity is too fast, this term becomes large. Now, what is this guy here doing? So this guy is adding a random Gaussian process. So what is a random Gaussian process? It's like every time you do the integration step, you roll a dice, right? But this is not a completely random dice. That if you collect enough of those rolling dices, the mean of the process is 0. So you're effectively not adding an effective force. You're always adding and subtracting. So this number is either positive or negative. But if you were to take the average over the course of a simulation, it should be 0. The mean should be 0. But then what about the amplitude of that Gaussian process, right? So you're getting a number at every time step. You have your pool, you get a number, but the distribution of numbers is actually given by this equation here. What this equation here is telling you is, or the way it is formulated, is that it not only depends on gamma, which is the viscosity, can anyone tell me what the units of gamma are? For us, by looking at this, it's 1 over time, right? And that 1 over time, if you want to think of it, is like the average time that it takes for you to have, like in a viscous media, to like the average time between collisions. Like every time, like you move, you collide here, you move, you collide here. So the amplitude, or like the sigma of that Gaussian process, is actually given by this equation, which also depends on gamma. So in reality, this is not 2 terms, this is only 1 term, right? And it also tells you that it depends directly on the temperature. So what you can actually do is set a temperature, and every time you run the simulation, you are guaranteed that by adding this single term, I call it a single term, your simulation is going to be limited to a particular set of temperatures. But now, what happens? I mean, you can think that this is, you can argue that this is very stupid, because you're actually adding a random term to a deterministic force field, right? What if the simulation becomes dominated by this term? It just becomes Brownian motion, right? So it has to be this gamma, which you can actually, when you go into simulation, you can actually select this gamma, the temperature coupling, has to be set to a small value. Otherwise, this term starts dominating the simulation, and the force field becomes irrelevant. So you want to have some temperature control, but you don't want the temperature control to be dominating the simulation. Also, there's an important detail here. This gamma is going to tell you over what period of time the temperature is being controlled. So like say you set gamma to 1/5 picoseconds. So that means that at a 5 picosecond period, your simulation, 5 picoseconds, your simulation on average is at a given temperature T, right? If you want to like control it very tightly, say I want every femtosecond to be at 300, so then you're going to end up with a very large gamma, right? And then this term becomes dominating the simulation. So you don't want that to happen. You want it to be a small value. You want the force field still to be dominated. You are only introducing this term or these two terms, however you want to look at, to control the temperature. But but there's the limitation that the temperature is only going to be controlled over the temperature coupling time that you use and you set up this gamma for. So typical values are 5 because 5 is already pretty big, but typical values are 1. For production simulations, you normally use 0.1. Is that so like 0.1 being every 10 picoseconds. So like over 10 picoseconds, temperature is regulated. So if you look at the instantaneous temperature, it's actually oscillating. We're going to look at that later. But if you average over 10 picoseconds, the temperature is what you set it to. So are there any questions? Or
the trade-off with that, you want to set gamma as small as possible, but why wouldn't you necessarily do that in some cases? There's a trade-off, I guess.
So why wouldn't you do it?
Right. So you want to set that number, I've heard, as small as possible.
Yes. So otherwise, so this is also making the system more viscous. So if you set it to a very large number, it becomes like the water is not water anymore, and things like this. So you want it in a way that you're still keeping the temperature close to a desired value, but you don't want it to set it in such a way that you're affecting the physical properties of the system of interest.
Okay, so yeah, you would determine certain physical properties.
You should, yeah. Ideally, you are interested in some physical property. Yep. Are there any questions?
What's the atomic basis for this drag?
This is just temperature control.
That's all.
Yes, yes. So in fact, you are introducing an artificial drag. But because you are doing it very softly, you can still say that the drag is negligible. So that's why you have to pick a small value. Otherwise, you're really you're effectively adding a non-physical drag. Yeah.
So are these equations calculated every 10 picoseconds?
These equations are calculated every time step.
Every time?
Yes. So every time step, you calculate the next step, you have to add this term. Okay? So I'm not going to go into this slide, but I will give you, and this is one of Klaus's slides, but the reason I like to show it is because if you think that the temperature control is complicated, the pressure control is even more complicated. But I want you to make sure, I want to just to like show you that it's formally derived, and it's also Langevin piston associated to the volume of the system. So where you actually control, here you have this term here, so you're looking at the fluctuations of the pressure, but you're doing that by changing the volume of the box. But let's not go too much into the details of this formalism. So are there any questions up to here? Like anything you guys want to discuss?
I noticed that it says here that you were to discuss the analysis of equilibrium and non-equilibrium properties.
We'll do that in a little bit. So is there anything in particular you're interested about analysis of equilibrium, or?
Nothing in particular. I just haven't heard much discussion of how you would model non-equilibrium processes in
Well, a simple one will be stir-MD, right? Like what we saw yesterday, where you pull. That's a classical non-equilibrium work, right? There is also for conformational transitions between like substates of a protein that all like say there is something called targeted MD, which it effectively what it does is it applies forces. Like you have two, like say you have a kinase, it has an open and a closed state. So you can actually apply forces in such a way that if you're in the open state, you apply a force that you bring it to the closed state. And because you're applying an extra work that's obviously near equilibrium or quasi-equilibrium. But you can do it back and forth. And that's also like, that can be done with the multi-copy algorithms. Like you can actually generate multiple of these transitions, and then you can, like at the post-process stage, you can look at like all this ensemble of transition states, or the transition ensemble, and you can, sometimes you can reconstruct the free energy associated with a transition. But you can do all of that out of equilibrium. But I don't think we're going to touch on that in too much detail, but it's certainly possible. So, well, we were discussing about the geometries of the simulation box. So for instance, this is a 100 million atom simulation. So when you start cutting corners, literally, from the simulation box, you start saving time. You start saving a number of atoms. And something that no one raised a question about, well, maybe, is like the term that the electrostatics are calculated. There is a sum over all particles in the simulation box, right? So you can imagine if you have 100 million atoms, you can imagine that you're actually calculating. It's an n-squared problem to do the electrostatics, right? In any case, it's possible. I'll give you some benchmark numbers just for reference. So if you know you're not getting anything like this for your system, then you know something is no good, because we actually get them for a very large system. So let's look at these examples here. So let's look. This is what we call a scaling curve. So on the x-axis, we have the number of nodes. This was done on the Titan. Actually, this is Blue Waters, which has a Cray XK7 or a Cray XE6. The difference between the XK7 and the XK6 is that the XK7 has a GPU. So we're talking about 16 of these nodes on Blue Waters. So if we have, for instance, let me see. So for instance, the solid triangles, at least here, the solid triangles correspond to the GPU accelerated. The empty triangles correspond to the non-GPU accelerated version. So what you can see is this is what we call linear scaling. And what that means is if you add more nodes, you still get an increase in performance that looks linear. Once you reach this plateau here, you're not in linear scaling anymore. And what that means is you really, if you keep adding nodes, you're not going to get any more performance. So let's look, for instance, to the microtubule, which is a 15 million atom system. It's right here. So if you look at it, it goes down very quickly. So if you're using something like 200, like around, I'll say like 400 nodes, you're getting about 8 nanoseconds a day, which is pretty good. In the case, let's look, for instance, HIV, which is 64 million atoms. You can go all the way. Actually, you can go all the way here to 16 nanoseconds a day by using 4,000 nodes, which is a pretty considerable part of the machine. I think what the story here is that even though like that very basic equations we were looking at before, but not basic, simple equations where we were looking before, they can still extend to very large systems. There are some intricacies when you extend to large systems. For instance, but you guys don't have to worry about that, is for instance, because of, like say this simulation box is very large, it's like can be easily 100 nanometers. So when you start having that large of a simulation box, your coordinates, your R, starts becoming large, right? And then you start requiring like larger chunks of memory to store those big numbers, right? And then you end up, when you're actually calculating the coulomb, you start adding like small numbers with big numbers, and then you have these risks of having overflow, or you like having, like if you don't have you're not using, say, for instance, double precision for those of here. I mean, I have conversation with some people here. Like if you don't use double precision, then you're gonna have problems where you introduce inaccuracies. So all those kind of details have to be taken care of, but they're taken care of by the programmers. The end user doesn't have to take care of that.
What about GPUs that have only single precision? Are you saying it's okay to use single precision?
It is okay to use single precision, but only because the programmers made that easy for you. But yes, you can use single precision without any concerns. In any case, for the end user, which are you guys, you can run very large systems and you don't have to worry about the intricacies of making it, all these equations work for these systems.
It looks to me, just from like eyeballing the graph and comparing the different simulations, that the scaling with n is roughly proportional to n?
The scaling with n number of nodes?
No, number of size of the system.
So like if you go from here to here, here to here.
Like like at 256 nodes, if I'm comparing the HIV to the microtubule, it looks like the change in n is change in system size is about a factor of 4, which is about the same as the change.
Oh, it really depends on the system. Yeah.
So that's what I was going to ask. Can you comment on?
So well, okay, all of these systems, they tend to behave similarly because they have a lot of water, so they're not really dense. So most of the actual simulation box is water. So they're not dense. So that's why they kind of behave similarly. If they were more dense, like say for instance, we don't have STMV here. The STMV is a very dense system because it also includes the RNA inside of the capsid. It has the capsid, the RNA. That one has a different. It behaves differently. So because in all these, actually all of these 4 systems, most of the calculations is water-water interactions.
And those are faster because?
They're optimized. Yeah. Okay. Oops. So this is Jim Phillips. He's the main developer for NAMD. This is what it looks like when he is tuning a NAMD run on a supercomputer like Blue Waters. So you see like what these are. These are events. Probably just looking at like a single time step where you have to load the trajectories. You have to load the coordinates in memory. You have to calculate the potential on all these. So tuning a NAMD for a machine the size of Blue Waters or Titan requires a dedicated programmer, and it requires like a lot of knowledge about the machine and about the algorithms. So if you notice what the main problem when you are working at these scales, you want the machine never to be idle, right? Like because you're transferring data from one node to another, and then if you're not careful enough, what you end up doing is you spend most of the time copying data from one machine to another. So the way that like Jim does this is by minimizing latency between the communication between the different nodes. Okay. So back in the day, actually this is not what I wanted to show this here. So for like a current reach of all atom molecular dynamics is in terms of compared to the size of the current simulations, if you can go from anything from very small molecules to very large complex systems, right? Like you can go from studying your alanine dipeptide in water all the way to simulating like a virus capsid. So that's that's how far molecular dynamics has, all atom molecular dynamics has come. Coarse-grained molecular dynamics is also still a valid option, but there seems to be the performance of all atom is allowing it to get closer. Yes?
So for virus capsids and larger molecules, you said there are millions of atoms. How about proteins in general? Like, is it for a 50 kDa protein, let's say, how many atoms can it expect?
For how many? 48 K.
Protein, yeah. Would it be 100,000 or would it
48 K, kilodalton, you say?
50 kilodalton?
That's like 500,000. Yeah. Yeah, roughly. So that's, well, I mean, that's well within the reach of current practices. Yes. Yeah. So the all atom molecular dynamics has go a long way, but we still have the sampling problem, right? We are still limited in how long the simulations actually, how long the simulations are run for. So this, as we mentioned before, the speed limit is the time step at the inner loops of the velocity Verlet algorithm. And actually, as we mentioned before, that's simply bond stretching. That's what happens in the femtosecond, like 10 femtosecond time scale. If you want to look at more interesting dynamics, then you have elastic vibrations. We happen in a 10 to the 3 picoseconds. Like if you want to look at hinge bending motions in a protein, they happen well into the nanosecond regime. And if you want to look protein folding, as we've seen before, that takes you a couple microseconds, depending on the performance you're getting. That can take like days to hours. If you're really going to look at more complicated motions, you're going to be looking at like local folding and folding transitions, or like rotation of side chains, like actually buried side chains. That's very slow. Those very slow processes will take years to calculate. So this is just by running straight molecular dynamics. So we still have the timescale problem is still still in the field. So there are as I mentioned before, there's the enhanced MD's methods, like you effectively can pick. There are some arguments that you can predict an order parameter, but you can explore the, like for instance, if you're interested in some rotation, you can force that rotation and then explore what will happen if you're let it equilibrate. So you go out of equilibrium, but then you slowly let it go back to equilibrium, and then you try to extract the information from that equilibrated path, which is actually a collection of multiple simulations. Okay. Let's. We have a few more minutes left, so let's talk a little bit about the format of the PDV files and what, I mean, I think everyone is familiar with the protein data bank. Yeah. Yes, so you know, like a typical PDV contains the atom names, the residue names, the residue ID, which is an integer, the coordinates, which are usually in fixed precision format, include the occupancy, the temperature factors, and the segment ID. So most of the hydrogens are missing. As you guys have learned, you can add them using PSFGen, which is in MD and BMD. I don't know if anyone has any questions about the PDV. PDV has multiple limitations, particularly because of the fixed size of the coordinate fields. So they currently was the PDBX was released. That's a new PDV format. They've been trying to push it for a year now. I mean, when we published the capsid structure, because of this size limitation, what happened is they split it in multiple PDV files. So they assign us something like 100 PDV IDs. But then when they introduced the PDBX, then we pushed them to to release a new PDB format. In the new PDBX format, there is a size limitation to the field of the coordinates.
So you can have more decimal places.
You have more decimal places, and yeah. So before it was 4.3, right? So which is crazy, but now the new PDBX is it allows for larger size molecules. Like that, the real problem is yes, you're limited to 4 integer, like 4 before the comma and 3 after the comma. So that's new in the it's called the PDBX. Support for VMD is on the way. There are not that many structures that require PDBX at this moment. I mean, I know our capsid structure requires PDBX, but not every structure requires PDBX. So in the future, as EM becomes more prevalent, we'll start to see more of these PDBX coming out.
Can you say what the temp factor is?
So we're going to talk more about that in the next section, but the temperature factor is effectively like how much wiggle there is in a crystal structure. We'll talk about that after the coffee break, which is, I think we can go for a coffee break at this point. But before that, I don't know if anyone has any questions.
I was curious if there were different methods for adding.
Sorry?
If there were different methods for adding the hydrogen atoms. I know that you can use the topology file, but I mean, it affects the results quite a bit, as I understand it.
It does. So, for instance, if you're trying to do, like say you're trying to reproduce some chemical shifts. Chemical shifts, that's a case where you actually need flexible hydrogens, because they actually capture those motions. So a more accurate way of placing those hydrogens is a prop Ka. You can actually, it has a better hydrogen placement. Yeah, as you said, when you use the topology file, he just fixes the hydrogen using the internal coordinates. Yes? So when you have a protein complex, right, you can have multiple segments, right? So the segment ID tells you that, like, say you have a 2 complex protein. So you have a 2 protein complex. So the first complex is assigned 1 segment, and the second protein is assigned another segment. So you can differentiate one from the other.
So you can split them to analyze them?
That's actually very true. You can actually, in VMD, you can select by segment name or segment ID. And so you can, name, if it becomes particularly useful when you have like a very large system, you want to be able to kind of navigate in the complex. And one way of doing that is by the segment ID, kind of more coarse coordinate. Mm-hmm. Yeah.
Kind of. You kind of just answered it, but I was just going to ask, is that the same thing as like the chain?
So they're different, yeah. Because, okay, so they're different. Yeah, chain is only 1 character, which is a big limitation. Segment is 4 characters, so that gives you more. You can have more segments than chains. So you can have 1 segment. Well, you can have 1 chain that is made of multiple segments. So chains are in principle larger complexes. It's kind of an outdated field, to be honest. The chain. Yes.
One thing that would be very helpful for me, and I don't expect an answer right now, would be a list of which parts of the record are actually used by NAMD, and which are just intended for like human consumption or for analysis later.
So NAMD usually only uses the coordinates, the coordinates, because and the occupancy and beta fields. But NAMD, you don't take those occupancy and beta fields from the PDB. He doesn't care. You use them more as flags to inform, like say,
To fix the protein structure.
Yeah, like you can tell him, okay, I want these atoms to be fixed. So you said these occupancies, but it's more of a flag. It's not really it doesn't have a real meaning. When in a PDB, okay, so maybe this is.
The atom name doesn't matter at all?
Actually, no. All it matters, because all he does is he reads so it's a caveat, but yeah, all he does is he reads a list of coordinates that has been pre-sorted before. So he gets the sequence and the atom names from the PDB. Oh, no, actually, he gets the sequence from the PDB. No, I take that back. He does get the atom names from there.
You wouldn't know the masses.
The masses are in the PSF.
Yes.
Actually, the masses are not even in the PSF. The masses are in the parameter files. The PSF has the charges. The masses are in the parameter
You don't
file.
know what's the coordinates. You don't know what the mass is.
The mass is associated with the atom type.
Yeah.
Mm-hmm.
All this stuff would be super helpful if it was on the AMD website somewhere.
I think it is there. Yeah, I think it's in the tutorial. Yeah.
Tutorial in the back. He should be telling you, don't you have a slide of the parameter field? Because you have to read the atom name, you get the atom type, and that's where the charges, everything were assigned.
I know it's probably on there somewhere.
It's supposed to be in the index. That drove me crazy, so I just had to put it in there.
It's difficult to do.
I understand.
We're approaching our way to
Yeah.
clarity.
Yeah, I mean, you guys also, so the PSF actually have the atom names, right? The atom types, so if you ever look at a PSF, we should probably look at it, has the atom types. So I mean, an actual atom can have multiple atom types depending on what residue it is in. As you mentioned before, the masses are actually associated with an atom type, right? And so is the charge. The partial charges. Yeah. So in the actual PDB, the occupancies, you can have multiple, like for instance, some crystals, they can capture 2 conformations for a sidechain. So you can say, they will tell you the occupancy of each conformer. And that's what comes in the occupancy field in the actual PDB. That's what it means for the PDB. And actually, when you're preparing your structure, you should take into account if it has multiple occupancies, because that probably means you have to study the 2 systems. Because it's telling you that sidechain can exist with a probability in this in a particular conformation and with a different probability in the other conformation. So it's not necessarily disorder. It's just it can sample the 2 states. Yeah, the beta fields, we'll talk more about that after the coffee break. But
The value of the occupancy then would be
it's a probability. So actually, you can get free energies from that occupancy, because you can also sometimes for ions. They also give you the occupancy of the ion, and it's not always 1, which means the ion may or may not be present in all the diffractions. So you actually can calculate a free energy from the occupancies. Okay, so I think we can have a little break.